Hey everybody, welcome back to Mem Makes. Today we're covering nutrition. Might be a little quick in today's video. I'm going to try and get through as much as I can in as little time as I can because I value everybody's time, including my own. So let's get right into it. Starting with the basics, then going through the rest of what you see there. Believe me, it won't be as hard as you think it is. Alright, spent too long last time I tried this video, so we're really going to speed through the basics. We got carbs, fats, and proteins, right? Carbs are 4 calories per gram, fats are 9, and proteins are also 4. Carbs going to be your basic building blocks or not building blocks, that's proteins. This is going to be your energy, right? Break down carbs for energy, break down sugars, mainly becomes ATP, and that's how we get most of our energy, right? Lipids, those are going to be used for energy storage as well as like waxes to waterproof stuff, steroids for hormones, and phospholipids for cell membranes. And then proteins, you've got basically stuff that builds literally everything, right? You got hair, collagen, muscle, bone, anything basically that's a fiber, that's going to be protein. As far as caloric needs, we've got a very fancy equation here opposed to generally using, or kind of guessing, as most people do. We got the scientific equation there to do it. Make sure you're using kilograms for weight, centimeters for height. I'm 5'8". I'm about 150 pounds. I put myself at a 1.4 for the active. I don't know. Technically, you're supposed to use like in between numbers, but like not as active as I'd like to be. However, I was being realistic. So my total calories in a day is gonna be 1759. So a little bit lower. It's gonna be different for everybody. General rule as far as macros go, you're going to want 60% of your macros, 60% of your calories in the day to be carbohydrates. So if I were to take that number, put it in my calculator, times 0.6, that would show me how many calories should be carbohydrates for me. Same with fat and protein. This is very general for the American population, or general population, I suppose. So if you're really trying to put on like a big bulk, you can kind of increase your protein a little bit, play with your macros a little bit, use your discretion. However, keep in mind that more than, like around 50, I believe, grams of protein at a time, your body can't really metabolize more than that. So eating astronomical amounts of protein isn't going to do as much for you as you would like, other than maybe give you a tummy ache. As far as proportions go, making it nice and easy, this is called My Plate. This replaced the food pyramid. Very easy to look at, very easy to understand. This should be everywhere. Show this to your grandparents, show this to your parents. Everybody should know this, because look how nice that is. Look how easy that is to read. You don't need to be a scientist or a doctor to understand what you should be eating based on this diagram. This thing is the gold standard. But yeah, we're at 3 minutes? Perfect. We're moving right along. But yeah, going back to carbs, right? Those are going to be 60% of your calories, because like everything you do requires carbs. Thinking requires carbs. Your brain requires carbs. Moving requires carbs, because it all requires ATP energy, right? Everything that's a carb gets broken down eventually into something that can be turned into ATP. Put an asterisk on that everything. But how do we get that ATP, right? What are the chemical processes that actually allow us to get that energy? That's what we're focused on today. So we're going to jump right into that. This is a note sheet that I made separately. I was going to do one for this, but going through remaking it all and all that was just going to take too much time. So why not just show you the work I already did? So we're going to be focusing on a couple things. First thing we're going to be doing is glycolysis. Some people call it, or it's also called anaerobic glycolysis, right? This is going to be the energy system you use when you're doing more than about 15 seconds of exercise, but less than a couple minutes. This is going to be like your track athletes, like your 400-meter guys, up to maybe like your 800, but after that you start getting a lot of lactic acid, and then you switch into some different metabolic systems. Anyways, right? Start with glucose, because that's the king of monosaccharides. Glucose gets turned into glucose 6-phosphate through the use of the enzyme hexokinase. Okay? This also requires 1 ATP right here, which turns into ADP. ATP, adenosine triphosphate, ADP, diphosphate. So you're losing a phosphate, burning some energy to get that glucose 6-phosphate. Eventually, that glucose 6-phosphate will turn into fructose 6-phosphate. And then we use an enzyme called phosphofructokinase, or PFK. And that will turn, as well as spending another ATP, that will turn into fructose 1,6-biphosphate. Fructose 1,6-biphosphate. A couple steps we skip because they're not as important to us here, other than one little addendum. There's an enzyme here called G3PD that produces 1-NADH, and that is important for the electron transport chain, which we'll get to eventually. The end of the glycolysis happens at step 10, where phosphophenylpyruvate turns into just pyruvate through the use of pyruvate kinase enzyme. And if you look here, we're actually gaining an ATP now, which is pretty cool. So through the use of this, we generate a little bit of ATP, but mainly what we generate and what's the most important is this guy right here, pyruvate. Pyruvate is going to go into our next energy system, which is called the pyruvate dehydrogenase complex. It's not really an energy system, I lied to you just now. It's more of a conversion system to get us acetyl-CoA, or I just abbreviate ACoA. But acetyl-CoA. Now that's going to be very important for us for the Krebs cycle, or the TCA, the tricarboxylic acid cycle. Very important molecule. But we get there through a couple of steps in this complex. We've got 3 enzymes, PDH, DHLT, and DHLD. Those 3 enzymes can be abbreviated into E1, E2, and E3. And then we got a couple coenzymes that also help with this, right? We need their help, but they're not necessarily the enzymes that are doing the work. So pyruvate binds to the TPP, this coenzyme, on E1. So on this enzyme, this guy lives there, and pyruvate binds to it. Then it becomes decarboxylated, and then it becomes rearranged, gains a proton, and then is transferred over to E2 on the lipoic acid, or the lipoene. So that's where this guy lives. He lives on E2, and he's the pyruvate, after being transformed, goes there. Then it gains an acetyl group and becomes acetyl-CoA, which is the important thing that we're looking for, right? We want that acetyl-CoA. And then it kind of fixes itself, making some more NAD and some more FADH2, which will go onto the electron transport chain later. So we get some really important little chemicals, little molecules here, mainly these 3 guys right there, ACoA, FADH2, and NADH plus H+. There are some things that can inhibit or activate this, right? Like if we're at rest and we have a lot of ATP lying around and we don't really need to make any more, that can inhibit it. Same with NADH, because like, why would we use this if we have a bunch laying around, right? That makes sense. It can also be inhibited by PDH kinase, which is another enzyme. If it phosphorylates the E1 enzyme, then it becomes inhibited as well. And then if that happens, then ADP and pyruvate can also uninhibit it, right? Because that's the presence of those burnt molecules where it's saying, hey, we need more energy, right? These molecules are the offspring of what happens when we're burning energy, so like turn back on. And then as far as indirect activation, same thing that I was just talking about there. Yeah, that basically covers that. So focusing on these guys here, right? We want these guys to go, we're going to follow them to the tricarboxylic acid cycle. Now bear with me, it was very late when I was studying, and I didn't really know where I was going with this sheet, so I kind of drew it in a circle. So we're going to flip this thing around, and we're going to look at the tricarboxylic acid cycle. So this is the Krebs cycle, also called the Krebs cycle, right? We've seen it before in all sorts of stuff, mostly like high school bio classes, right? It's going to start with acetyl-CoA and this stuff called oxaloacetate. They're going to mix together using the enzyme citrate synthase, and they're going to become citrate. Now that's step 1, right? Step 2, citrate becomes isocitrate through aconitase, which becomes then alpha-ketoglutarate through the use of isocitrate dehydrogenase. And notice here, we gain 1-NADH, and that's circled for a reason. You can see that there's an arrow that it goes to. That's going to the electron transport chain, as I've said before, right? Moving through, we got alpha ketoglutarate using alpha ketoglutarate dehydrogenase becomes succinyl-CoA, gaining another NAD or NADH. We're also expending some CO2 here, so that's where a lot of your like exhaling, right? You're exhaling CO2. A lot of it comes from this cycle. Succinyl-CoA uses succinyl-CoA synthase to become succinate. And here we're actually gaining an ATP through the generation of a couple other molecules. So you can see there's two arrows there ending in ATP, which is pretty cool. Succinate becomes fumarase through succinate dehydrogenase. Succinate dehydrogenase. Shout out Eric Sandler from Brockport, which is where I went to college. Dude's a legend. One of the dudes that made me want to become a professor. He's awesome. He had a way of saying this word that just made it stick in my head forever. This reaction is also going to get us some FADH2, which will go to the electron transport chain, as I've said before. But succinate becomes fumarate through succinate dehydrogenase. Fumarate becomes malate through fumarase. This requires some water as well. And then that malate is going to go all the way back up to the top of the Krebs cycle or the tricarboxylic acid cycle, plug in to that and become that oxaloacetate, which plugs in with the ACoA, starting the chain all the way back over again, right? So it's a circle. I didn't draw it that way, but the whole thing's a circle. You're going to see that in most diagrams, right? The circle all does, it keeps cranking, keeps taking that ACoA, turning it all the way around, generating those NADs, those FADs. Those all get sent to the electron transport chain, which is right here. There's a couple of different complexes for the electron transport chain that we're going to focus on. Complex 1, 2, 3, and 4. And then there's also, like, I don't know if it's called complex F and complex G, but they're like little subcomplexes that also happen. But we can see here that NADH that we were following from all the way through, right, comes right into complex 1 here and has an enzyme, NADH-Q reductase, right? That enzyme is going to generate 4 protons. That's very important because a gradient is going to occur with these protons, right? There's going to be an unbalance of protons. There's a proton imbalance. Now that NAD can enter here through, depending on where we're at in the body, right? If we're in the muscle, or I'm pretty sure that's supposed to be the liver. I'm pretty sure. Let me double check. Memphis, make an edit somewhere about this. Clarify where those are in an edit. Okay, I was pretty much right about what I was saying. So this guy right here, he does the NADP or NADH into the mitochondria from muscle, and this guy right here does it from the heart and the liver. You were pretty much right, Memphis. Back to you. However, one of these is going to bring you to the heart, and one of them is going to bring you to the liver or skeletal muscle. I don't know. Memphis, do the thing, please. But yeah, that's complex 1. At the end of the day, it's going to transfer electrons from some iron to this stuff called ubiquinone. Ubiquinone is going to be important. It's going to come up a lot. It also, Q is ubiquinone, just in case that comes up. Complex 2 is where we go next. Complex 2, this is where FAD enters, specifically on the G part of complex 2. That's going to have the enzyme G3PQ reductase, and that's going to be the shuttle that gives like your electrons and your FADs and things to your skeletal muscle. This is where FADH2 becomes FAD, and yeah, so that's the G part. The C part, though, the actual number 2 here, transfers electrons from that FADH2 that we're getting from the electron from the Krebs cycle to this stuff called ubiquinone. Now here, C3, we move on, and this is going to be the enzyme ubiquinone cytochrome C reductase, right? Now this is going to generate 4 protons. Notice step 2 did not generate protons. That's the only one that doesn't, number 2. But complex 3 generates more protons, and it moves some electrons from cytochrome B to cytochrome C. And then finally we're moving into complex 4, where we got cytochrome C oxidase which just moves those electrons onto oxygen, making us some water and generating us some protons. Now, what those protons are going to do when they're generated is they're going to flow out into like the outer membrane of the mitochondria, right? Didn't mention this, but all of this is taking place, at least all of the electron transport chain is taking place in the inner membrane of the mitochondria, and then the TCA cycle is taking place in the matrix of the mitochondria, right? We always knew they were the powerhouse of the cell. This is how they do it. So now if we look here, it says all of the protons, where are we? All the protons that were generated with the gradient, or cause a gradient, which uses an enzyme ATP synthase, and when that enzyme uses that gradient and causes it to flux, we get a massive amount of ATP. And that's where we generate like just boatloads of our energy. This is where, all of the ATP comes from. It's pretty cool. Now we can have different inhibitors to our different complexes in the electron transport chain. So our inhibitor for complex 1 is going to be called rotenone. Our inhibitor for complex 3 is called antimycin. And our inhibitors for complex 4 are carbon monoxide. See how there's no 2 here? It's just monoxide. So that's going to be like the bad stuff that you always breathe in. You got the detectors for in your house, right? And cyanide, which comes from like apple seeds, and it's like a poison, whatever. There's also ADP to ATP translocase, which moves stuff in and out of the mitochondria, and that can be inhibited by plants and like mold and stuff. So like that's why we don't want to breathe that stuff in. But yeah, we really just followed a glucose all the way through its entire metabolic life through the electron transport chain, watching all of the ATPs it generated. The last thing we got to talk about, though, is the pentose phosphate pathway, because there are some cells in our body that don't have mitochondria. Can you guess which one they are? I'll pause for a second for you. What cells don't have mitochondria? That's your question of the day. I'm going to start doing that. Question of the day. Something that I... Don't look at the screen. Don't look at the screen. Question of the day. What cells don't have mitochondria? 3, 2, 1. Red blood cells. Red blood cells don't have mitochondria because of the way they're shaped and because they transport oxygen, right? They got those, they're made of 4 little heme proteins, I believe, hence why it's called hemoglobin. And they carry iron, and so they don't have mitochondria, so how do they keep themselves alive? How do they make sugar? That's going to be through the pentose phosphate pathway. So this is what allows them to make NAD, and they make ribose, which is used for DNA synthesis, right? So they still need fatty acids and other things. They still need fatty acids and other things, and they still need to make DNA because they still need to be able to do their own stuff. So they go through this. Step 1 is the redox step, which is irreversible. In the liver, NADP is used to in the GT pathway, and it prevents O2 damage, right? O2, free radicals, they float around the body, stealing electrons, attaching things, causing little H's to float around, little protons, which is bad. We don't want little floating guys. We want all of our chemicals to be together and happy and in a family, right? So when they're out there, they're called free radicals. They're bad. We eat antioxidants, blueberries, things like that to get rid of them. So this is helping with that. It's preventing that O2 damage, and it's also synthesizing stuff. In red blood cells, it's only to prevent that O2 damage, not to really synthesize stuff as much. Yeah, I lied to you earlier. See this? This didn't happen in red blood cells. This part right here only happened in the liver. Watch yourself, Memphis. Step 2, interconversion stage, which is reversible. Okay, that's very important. The reason it's important because if it wasn't reversible, we wouldn't be able to make R5P, ribose 5-phosphate, for making like DNA and stuff. And without that, there would be no reproduction, there'd be no protein production, and then the cell would die. So that's bad. We don't want that. So during this interconversion, they can make R5P, and then if they don't, they can make all the other sugars. They have epimerase. Epimerase is an enzyme that causes these guys to be able to switch back and forth as needed. That's R5P and X5P. And then there's isomerase, which changes the keto R5P to aldo R5P. One of those two. K and A. That's fine enough for now. K and A. You'll probably know it if you're studying this stuff. If not, it won't be the end of the world. But yeah, that's literally all of the different pathways that we're worried about covering today. We did it in just under 20 minutes, so that means the video is gonna be 21 minutes long. We'll take that. Yeah, that covers it all. If you guys like this kind of stuff, let me know. I can either go more into the basics and talk more about like the general nutrition stuff, or we can dive more into the science and I can explain some more of this stuff. But that pretty well covers all of what I was looking to do today. If you have any questions, let me know. If you guys have any ideas or things specifically that you want to talk about, let me know. I really enjoy talking about the human body. I think it's the most fascinating and perfect machine. And so, yeah, if you guys are curious about anything, let me know. Things coming down the line, we're going to have an endocrine video coming out soon. And then after that will most likely either be an anatomy video or a neurology video, both of which will be fun to make. The anatomy video will be definitely a bit longer and will be a bit tougher to do. The neuro video will be super cool, but will also be pretty long. Yeah, keep me updated on what you guys want for like video length formats. I can do more crash course type stuff where I don't go nearly in as depth, or I can keep it a bit longer. I don't know if I really want to push past like a half hour for video length, because then like it becomes just like a full professor lecture, in which case why wouldn't you just go watch one of those, right? I know some people don't have access to those, but you know, whatever. Till next time, folks, lads, ladettes, we'll see you soon. Keep that brain working. I'll come up with an outro sooner or later. In the meantime, catch you in the next one.